Okay, you guys ready? How does an alien count to 23? How does an alien count to 23? Nope, no ideas? On his fingers. They have great ideas, and then they have some other jokes. Where do crayons go on vacation? Where do crayons go on vacation? To the Wax Museum. Okay. These are unfiltered, unedited, okay, uncensored. It's better than the internet, right here. Okay? Okay, the first thing I want you guys to talk about. Pair up. And I want you to have a conversation with your partner, your neighbor. Why do we have differences in skin color? Okay? Why do we have differences in skin color? Okay, I'm going to bring you back in. Some of you are having an awful lot of fun with this question. So I'm not sure we're talking about the same thing. Actually, it is a science question. It's not a sociology question. Any ideas, any guesses? Sir?
As we migrated away from Africa, we had no longer a stress to produce melanin in our skin. So when you walk away, migrated away from the equator, their bodies stopped producing as much melanin as lighter skin.
Okay, so the concept is around migration away from the equatorial plane. Okay? And the further away you get, the less likely you need high amounts of melanin. Why wouldn't you need as much melanin? What's that?
Less sun exposure?
Less sun exposure. Okay. Does it have only to do with the sun? Maybe something that, yes?
Blending in with your environment?
Blending in with your environment. Okay. Maybe. Okay. So you guys are getting away from the science behind it, and you're getting more into the sociology behind it, right? Or pressures, maybe. So where I want to go with this is there are about 1,000 to 2,000 melanocytes per square millimeter of skin. Per square millimeter. Okay, there's about 10 millimeters exactly. There's 10 exact millimeters in a centimeter. Catch that? There's 2.54 centimeters in an inch. Those of you that are metrically challenged. Okay? So there's a lot of melanocytes. That's in all of us. It doesn't matter what part of the world you come from or where your ancestors came from. You have all this approximate same, that's a big range though, 1,000 to 2,000. Right? That's like saying I scored between a 50 and 100% on the last exam. So that's a big range, but we all have the same parts. We all have the same number of melanocytes, about 1,000 to 2,000 per square millimeter. But the production or the activity of those melanocytes to produce melanin varies, or melanin, depending on how you pronounce it. And melanin is that dark brown pigment. It's a protein that's produced that's a UV blocker. So the problem with the sun, or the challenge with the sun, is lots of sun exposure means lots of UV rays. Now, I just got done talking about the last time we were together how you manufacture vitamin D. And I know that some questions have come up in SI sessions, and by the way, I want to make sure that I'm encouraging you to go to SI this week. I know that last week we had an exam, so SI attendance was lower, but it is one of the best ways to impact your grade in a positive way. And in the SI sessions last week, a lot of questions came up about this reaction. What do I need to know? Well, you do need to know that the sun is actually important. It's not all bad. In fact, it's one of the keys to the reason that we have life on this planet, and that we can exist as an organism. Because we need to actually manufacture lots of products as a result of the sun. Not just plants, but we. So like plants, we actually rely on the sun to help us catalyze this reaction to make vitamin D, vitamin D3, calcitriol. You can get vitamin D out of your diet. Usually the food source is vitamin D2 instead of vitamin D3, but they both can be precursors to create vitamin D. Why is vitamin D important? It helps you to extract calcium out of your diet. You need calcium for strong bones. That's not a lie. It wasn't just dairy farmers making something up. And we're going to talk about bone architecture in the next section. But this is how the skin is connected to the bone. Remember, we compartmentalize the anatomy and the physiology arbitrarily. We, as scientists, as the field, as professors, for students, we put it in this nice, clean chapter. And we say, this is what we're talking about the skin today. Does the skin work independently of the other systems? No. Absolutely not. No. Okay? And the skin and the bones are intricately connected. So the skin isn't going to say, I don't want any UV light to come through because sun needs to come through. So you need to have the ability to change the shade of the skin to allow certain amounts of sunlight, UV light to come through in order for this reaction to take place. But there's always too much of anything, right? So UV light can also be harmful, and we'll talk about that later in the lecture today. When the skin gets too much UV radiation, or when any cell gets too much radiation, you can actually cause damage at the level of the DNA. And if you cause a mutation at the level of the DNA, then you alter the programming of that cell and it can become cancerous. If it becomes cancerous, that's not good for us. And so we need ways to protect or block the UV light. So, our skin, melanocytes produce melanin, which is that natural sunblock. So the theory that was brought up in the back, which is pretty close to what we believe currently, there's a lot of sociology involved in this, but what I want to extract out is really the physiologic mechanisms. So if you look at this map, okay, this is an older map. It was updated in 2007, okay, from its 2004 original publication. You can see it came out of an anthropology publication. This is not an anthropology class. But what's interesting from these other disciplines is if you look at the distribution of indigenous people around the globe, around that equatorial plane, which is the darker colors on the screen, you have the highest amounts of melanin production in those individuals. And as you move further away from the equator, the intensity of the sun does actually get less. And so your ability to actually extract UV light in a beneficial way goes away, because you don't have as many hours of the day of sunlight. And so it would not be as advantageous for that group of people to have darker skin colors and block a lot of that UV light if they're trying to make sure that they're getting enough UV radiation in order to produce vitamin D3. So it does appear that there is a correlation. Is there a direct cause and effect? No. Okay? Are we trying to talk about any sort of anthropological migration of people? No. What I'm trying to explain to you is within the human population, we have a wide diversity of skin colors, and it benefits the species to have that based upon where these people came from. Okay? Is it as much of a deal today? No, because we have technology. We have more shelters. We have development that's taken place across all aspects of the globe. However, we still have this diversity within our patient population. And where it becomes interesting and how you might see it in the future is if you're treating a patient, okay, and you're treating a patient that you're worried about vitamin D because you're worried about this older postmenopausal female who had a hip fracture, and you're worried about their bone health, would the amount of sunlight that they get on a daily basis make a difference?
Yes.
Absolutely. Would their ethnic background possibly make a difference? Yes, it will. Okay? So those are the things that you need to be aware of, not because we really care about this map so much. I care that you understand that every one of us is going to have a unique background, a unique genetic profile, a unique ethnic background, and that's going to be your patient population. And so understanding how that difference is going to manifest itself in patients in the future actually will make a difference.
So people who are albino, have albinism, is that a cellular problem, or is that in the skin itself as the melanocytes start making the melanin?
So yes and yes. It's a cellular issue, and it targets mostly epidermal tissues. But it's not just at the level of the skin, like the hair loses its pigment too. Okay? Did you have a question earlier? Or are you good for now?
I have a question. Does that mean that maybe as people live more and more indoors, just like naturally everyone becomes lighter skinned as generations go by, of people just avoiding the sun more and more?
That's a hypothesis. I think no one's going to be around that many years to test it, but could be. I don't know. I don't know the answer to that. But it's an interesting thought. So let's move on to our second layer of the skin, the dermis. So the dermis, this is a deeper layer than the epidermis. Epi meaning on top. That's the superficial most layer. This underlying layer is very strong and it's very flexible. It's mostly dense, irregular connective tissue. Remember, the irregular type is the type that goes in all these different configurations. So it's strong in all directions, and you can kind of appreciate the waviness of these lines. They're not oriented all in one direction. It's made up of cells and other types of molecules like collagen protein, elastin, reticular fibers. Here are fibroblasts and macrophages under the cellular category. And it has a tremendous amount of vascular elements, or it's vascularized. A lot of blood vessels in the dermis. The epidermis doesn't have many blood vessels, but none at all. The dermis is where all the blood vessels exist. And those blood vessels here can actually, by diffusion, supply nutrition to the epidermal layer. That means that it goes from an area of high concentration to an area of low concentration. What is that? Well, that would be oxygen. Okay? Carbon dioxide can be removed. Waste products can be removed as they diffuse away from areas of high to areas of low. The other thing that's interesting about the vasculature in the dermis, in relatively no vasculature, or what we call avascular in the epidermis, is that the diffusion distance of oxygen is about 250 microns in tissue. So what that means is the tissue that's 300, 400 microns away is at risk of being underperfused and becoming what we call ischemic. And so when it becomes ischemic, usually it'll signal itself to slough off. And you know, we just left the epidermis, and you know that's exactly what happens. Every 6 to 8 weeks, you slough off a layer, and you regenerate it on a regular basis. And part of that reason for it is because of the fact that it's not vascularized, and your skin only gets to a certain thickness. Isn't it interesting that your skin today, other than calluses that we talked about, but your skin today is not really any thicker or thinner than it was when you were a kid? It's pre-programmed. How is it pre-programmed? It's pre-programmed because of the ability of oxygen to diffuse to a certain limit within tissues. Well, we also see a lot of sensory receptors here. You studied them a lot in lab. We'll cover a few of them here. But these sensory receptors give you tactile information, mechanical sensation, pressure, and even pain about your outside environment. Yes, question?
So when every 6 to 8 weeks when your skin sloughs off, is that the entire epidermis?
Yeah.
The entire?
Everywhere.
So then the dermis becomes the epidermis?
No, the dermis doesn't change. Remember, last week we talked about, this is a good question, last week we talked about the bottommost layer of the epidermis is called the stratum basale. That's the regenerative layer. That's the layer that turns over. But you never turn over the dermis unless you actually have a wound that we'll talk about later. Okay? So here's a different stain showing the delineation between the epithelium, and here is that nice line that I was tracing earlier in that previous slide. You can kind of see how it meanders around. And then the dermis. The dermis actually has two major layers. It has what we call this papillary layer, and then this reticular layer. The reticular layer is where you have a ton, scientific term, right? But you have most of your dense irregular connective tissue in your reticular layer. The blood supply is located throughout the dermis, both the papillary as well as the reticular layer. And the whole point here is to maintain structural integrity, house sensory receptors, so that you can actually interact with the outside environment. If we look at the two layers, we look at the first one, known as the papillary layer, they form what are called the dermal papilla. And the dermal papilla are these portions of the dermis that raise up. Do you see? The little bumps, the little humps, exactly. See right here, there's one and then two. Here, there's one right here. Now, you actually have an inverse hump that comes down here, and comes down here, and comes down here, from the epidermis. So the epidermis has these ridges that come down, and the dermis has ridges that come up, and it fits together kind of like an interlocking puzzle. The ones that come up from the dermis are called the dermal papilla. The ones that come down from the epidermis are called rete pegs. Two words, R-E-T-E, first word. Second word, P-E-G-S. Those are epidermal projections down into the dermis, and the dermal papilla are dermal projections that move upwards. Okay? That means that you have sort of this tongue and groove-like locking mechanism between the two layers of your skin so that they don't tear off of one another. The reticular layer down here is your dense, irregular connective tissue like we talked about. Lots of collagen fibers that are here, and it provides strength in all directions. So there's lots of white space to be writing all of these kinds of notes that I'm just mentioning, because none of that stuff's on that slide. Those are the kinds of questions that I've been entertaining since last week, when students come in and say, how do I study? So did you ever notice that sometimes there's a tremendous amount of white space on my slide?
Yeah.
And I'm up here jabbering away, and I'm reading stuff off of my lectern? These are the details that I want you to put down in there that I'm only giving you in lecture. Okay? All right, here's another stain. This one's actually my favorite. Why? Because I think it nicely shows these dermal papilla coming up, and this is what coming down? The pegs, the rete pegs. Very good. Okay, so you can kind of appreciate between the blue and kind of the pink this interlocking tongue and groove style like mechanism that holds a lot of strength for the skin. The papillary layer up top is made up of areolar tissue. It's more open-like. We talked about that in the last unit. The reticular layer down here is this dense, irregular connective tissue. And you can kind of appreciate the dense stain is a darker blue, and the areolar stain is more open, so it actually is a lighter blue color. So on this one slide, you can see the epidermis, and here's the stratum corneum up top. You can see the lighter blue in the dermal papilla of the areolar tissue. You can appreciate the rete peg coming down. You can label that right there, draw an arrow just for example in that white space to that rete peg. And then down here, you can actually see that the darker blue stain is your dense, irregular connective tissue. So it gives you a lot of architectural characteristics about the entire skin in one section. Okay, so what else is found here in the skin? Well, this is kind of the miscellaneous bit. We're going to talk a little bit more about some of these other things here in a moment in the lecture, in part 2. But here's where we're just going to identify them real quickly. So if we look at hair follicles, the hair follicle itself dips down from the epidermis, right? So the hair regenerates. The hair is a sort of modified component of epidermis.
So it's completely surrounded by epidermis.
It is.
Oh.
So the epidermis comes down to the hair shaft, and at the bottom portion of the hair is kind of like the stratum basale layer that regenerates skin. It regenerates hair. If you pull a hair out, there's another hair that will eventually grow back. Okay?
Are follicles found in the pegs or are they something completely different?
They're completely different. Yeah, they're not necessarily. The question was, are hair follicles found in the red apex? And the answer is no. They're completely separate. Sometimes they could. A lot of times they won't. Okay? So, for example, this is the hair shaft right here, and there's no appreciable red apeg coming down. But that's just arbitrary. I mean, sometimes it may show up there, and sometimes it may not. So we see hair here. We see sweat glands. Right? We see sweat glands found in the dermis. So the hair follicle is located at the level of the dermis, but it's an extension of the epidermis coming down. That's why we're talking about the epidermis. Sweat glands are located in the dermis. Blood vessels, nerves, and these erector pili muscles, or piloerector muscles. So these piloerector muscles, we call them kind of vestibular characteristics. That's a word that you've probably heard in other biology courses, where in other species, they serve certain functions, but now in mankind, they don't really have the same function anymore, and they may not even be utilized. But we have these remnants of the technology that's used in other technology, being biological technology, that's used in other types of species. So the piloerector muscles are the ones that actually make the hair on your limbs stand up, like when you get cold or frightened or scared. They're smooth muscle. They're under autonomic control. It's a reflex, and it's the same reflex in avian species that allows the avians, the birds, to actually fluff out their feathers. Okay, and they fluff out their feathers because by fluffing out their feathers, it creates airspace, and that traps warm air, and that actually keeps them warmer. Okay, that's why your goose down jackets, your puffy jackets that have goose down in them, that's why they're so warm, is you have all of these micro cavities of trapped warm air that is creating an insulation between you and the outside environment. One second, there was a question back here, then I'll come to you.
I just wanted to ask, does the epidermis surround the hair follicle?
Yeah, I'm going to show a hair follicle in a couple of slides, but the epidermis layer comes down, and the bottom portion of the hair is actually contiguous, if you will, or same embryonic material as the stratum basale and the epidermis.
Okay.
Okay?
Yeah, actually, I have a comment to make. How you said that how birds have erector pili muscles also that help their feathers go out.
They're called something else, but it's the
Right,
same.
right. Well, in humans like us, we don't have any control over when our hairs stand up. But birds, I know because I work with exotic birds, they can fluff up their feathers anytime they feel like it.
Yep.
They have control.
They
over
do.
that.
Wouldn't it be cool if we did? We'd be like,
Yeah, we know that they do.
that'd be weird, huh? Okay. Let's go through some review questions before we move on. All right, so show me on this picture, consider where keratinocytes first appear in the epidermis. Answer the following question. Which stratum is the first to contain dead or dying cells? A, B, C, D, or E? How many of you say A? How many of you say B? How many of you say C? D? And E. Okay, and how many are not playing? Yeah, I see a lot of hands not playing. Okay. So the first layer that contains dead or dying cells is which layer? A. What layer is that? That's the stratum lucidum layer. Right? Why is it dead or dying there? Because that's where it's absent of organelles. That's why it's clear. Now, one could argue that the granulosum layer is where it begins to die, but they're not dead. You're still creating products. You're still manufacturing keratin. Right? Keratinocytes are still alive. You're still manufacturing filaggrin in that granulosum layer. You're still manufacturing elidin. You package them in content vesicles, and you might use them in subsequent superficial layers, But the stratum lucidum layer is the first layer we consider to be a non-living layer. Okay? All right, next question. Papillary layer is labeled what in this picture? And it's made up of what type of tissue? Now I understand on the exam I don't have F and G options, but we can use them here. Okay, work together. This is a two-part question. Some of you felt that the two-part questions on the exam were tricky, so we're going to practice the tricks so they're not so tricky. Okay, who's got a defendable answer? Who's got a defendable answer here? In the very back. What was it? E is in echo? Yes, it is E. Why? Because it is? Explain it to us. Why is it E? Why is it 2 and areolar? I can barely hear you, man. You got it. It's above what? It's above reticular. I agree. Okay? That's all you got. All right. Well, it is definitely areolar, right? That's what makes up the dermal papilla. Right? That areolar. It was that lighter blue stained color in my favorite histology image. And the darker blue stained color down low was what type of tissue? Dense, irregular connective tissue. Okay? Very good. All right, we're going to move straight into the next lecture.